We're going to take a look at the synapse between the autonomic postganglionic neuron and the effector organ, autonomic regulation, and then comparing the sympathetic and parasympathetic nervous systems in this lecture. So the autonomic neuroeffector junction is just the synapse between the postganglionic neuron and the effector organ. So we're going to change names here. So instead of calling the end of the postganglionic neuron the axon terminal, we're going to call it a varicosity. Okay, so we've just got a different structure here. So let's take a look at a picture. So the postganglionic autonomic neuron, when it comes into synapse with its target organ, notice that we don't just have an axon terminal, and we do have some collateral axons here, but each of these collateral axons that's branching off here, they each have these swellings, and there's several of these swellings here, and each of these swellings is called a varicosity. And each of these varicosities will actually release neurotransmitter. So that's really different from what we were talking about before. So remember the neurons we were talking about before, they would come down and synapse with whatever their target is, and they would just have this axon terminal where they would be releasing the neurotransmitter from. But here we've got these swellings, and from each of these varicosities, we're going to be releasing neurotransmitter from each of these. Okay, and so that would happen along each of these collateral axons. So having these varicosities will allow the autonomic postganglionic neuron to release lots of neurotransmitter over a widespread area over the effector organ. And if we take a close-up look at one of these varicosities, notice that we've got some voltage-gated sodium and voltage-gated potassium channels. Remember, that's going to produce the action potential. So as the action potential comes down the axon, it hits the varicosity, and the action potential, remember, is going to depolarize the inside of the varicosity here, and that'll trigger the opening of these voltage-gated calcium channels. And that will make them open, and calcium will rush in to the varicosity, and that'll trigger the exocytosis of the neurotransmitter from these vesicles. So we'll release the neurotransmitter from the varicosity and onto the effector organ. And then the action potential will continue on through the varicosity because you still have some voltage-gated sodium and potassium channels in the varicosity, and it'll continue on down the axon to the next varicosity, and then we open more voltage-gated calcium channels, release more neurotransmitter at that next varicosity, and then we send the action potential onto the next varicosity until we get to the end. So that's just a little bit different arrangement than the other neurons that we were talking about before. So we see these varicosities just in these postganglionic autonomic neurons. That's where we find these. All right, so we just talked about this. This is just all written out here. And let's just take a look at another picture. In the picture on the left, we have an action potential coming down to the varicosity in this postganglionic neuron that's synapsing with this effector organ, whether this is cardiac muscle, smooth muscle, a gland, or if this is a sympathetic target, it could also be adipose tissue. So once the action potential hits this varicosity, that'll trigger the opening of the voltage-gated calcium channels. That'll let calcium into the varicosity. That'll trigger the exocytosis of the neurotransmitter. Neurotransmitter gets released and diffuses across this little synaptic cleft here, and then binds to its receptor on the effector cell and then has the effect. So is this postganglionic neuron that we're looking at, is this from a parasympathetic or sympathetic postganglionic neuron? What do you think? So the way you would tell this would be to look at what neurotransmitter is being released. So we've got acetylcholine, and then also take a look at the receptor that acetylcholine is going to be binding to on this target organ. We've got a muscarinic cholinergic receptor. So that tells us that this postganglionic neuron has to be a parasympathetic postganglionic neuron because they're going to release acetylcholine, and acetylcholine will bind to muscarinic cholinergic receptors, which will be found on the parasympathetic target organs. And then to get rid of the acetylcholine, because once we get our response in the target, then we want to get rid of the acetylcholine. And so acetylcholine will be chewed up by the enzyme acetylcholinesterase, which is right here in the synaptic cleft. So acetylcholine doesn't last long. It gets chewed up pretty quickly. Okay, and then the picture in the right, we've got another varicosity in this postganglionic neuron. Action potential comes down. That'll trigger the opening of the voltage-gated calcium channels. Calcium comes in, triggers the exocytosis of the neurotransmitter, and our neurotransmitter diffuses across the synaptic cleft to the receptor on our effector organ, and then we'll get a response in our effector organ. So this postganglionic neuron that we're looking at on the right, is this from a parasympathetic or sympathetic postganglionic neuron? So hopefully you said that this is from a sympathetic postganglionic neuron. And we know that because we're releasing the neurotransmitter norepinephrine, which is what the postganglionic sympathetic neurons will release, norepinephrine. And then norepinephrine will bind to adrenergic receptors on the sympathetic target organs. Okay, so that's how you know which one this is. So this is a sympathetic postganglionic neuron. And then again, we can get rid of the norepinephrine by a couple of different ways. It takes us a little bit longer to get rid of the norepinephrine, so the effects are going to last longer. One way to get rid of the norepinephrine is to reuptake it into the varicosity. And then it gets chewed up by the enzyme MAO, which is on the surface of the mitochondria. There's another enzyme called COMT, which is located out here. And that can also chew up the norepinephrine. And then the third way to get rid of the norepinephrine is the norepinephrine could just diffuse out of the synaptic cleft and then get taken away by the bloodstream and then join with the epinephrine that's been released by the adrenal medulla. All right, so regulating the autonomic nervous system. Remember that most of the organs in the body have dual innervation, just meaning that they're going to be innervated by both parasympathetic and sympathetic nerves. And what I want you to remember is that these organs are continually receiving signals from both the parasympathetic and the sympathetic neurons all the time. Okay, so it's not like you're turning one on, turning the other one off. Okay, so they're both always active. But what happens is that the body will adjust the activity of the parasympathetic and the sympathetic neurons. So if you're resting and digesting, then you're going to increase the activity of the parasympathetic neurons and decrease the activity of the sympathetic neurons. But if the bear jumps out at you and you've got to run away or fight, or if you're stressed for some reason or you're exercising, then sympathetic neurons are going to increase their activity and parasympathetic neurons would decrease their activity. Okay, so because both sympathetic and parasympathetic divisions are always active, we call that autonomic tone. Okay, so It's not like you're just turning one on, turning one off. They're both always sending signals, but you just adjust their levels of signals that they're sending. Okay. All right, so the autonomic nervous system is mostly under involuntary control, so you don't have to think about it. It just automatically does its thing, which is nice. So you don't have to think about, oh, it's time to, have my heart contract and pump the blood around. But you can have some conscious control over the autonomic nervous system with your cerebral cortex. So if you do think about it, you can slow your heart rate down. Okay, but you can't stop it, because your autonomic nervous system will say, no, you got to keep your heart beating. Okay. And then we do have our hypothalamus and limbic system. Emotions will affect the autonomic nervous system. So, for example, when you're angry or fearful, that will stimulate your sympathetic nervous system, and that'll increase your heart rate and blood pressure, for example. The brainstem in the pons and medulla oblongata, there are some autonomic control centers there. So there's a cardiovascular control center and there's a respiratory control center there as well. And then spinal cord, we do have some reflexes going through there for the autonomic nervous system. So these autonomic reflexes, remember we talked about that before. You could classify reflexes as being either somatic or autonomic. Remember the somatic reflexes, they innervated skeletal muscle, whereas the autonomic reflexes will innervate the smooth muscle, cardiac muscle, or glands. We can also call the autonomic reflexes visceral reflexes, so sometimes you'll hear that just because you're talking about the internal organs a lot of times. And we have lots of negative feedback loops going on to help us maintain homeostasis as well. Okay, and then this is just showing you where some of these important autonomic control centers are in the brainstem. So here's the pons, and here's the medulla oblongata that then joins in with the spinal cord. And in blue, we've got the cardiovascular control centers. They're going to control your heart rate and blood pressure. And then we have these respiratory centers, which are going to control your rate and depth of breathing. Okay, here's an example of an autonomic or visceral reflex. So this is how you control your blood pressure. And we're going to talk about this reflex again when we get to chapter 14. And then here's just another example of a autonomic reflex. So this one's just showing the intestine here, and we've got a sensory neuron sensing the stretch, and it will send a signal up to the spinal cord. It'll synapse here on an inner neuron in the spinal cord, which will then synapse on this autonomic neuron, which will then go out to the smooth muscle in the wall of the intestine, and that will affect the motility of the intestine. So that would be classified as an autonomic reflex because you're innervating the smooth muscle. All right, and then just comparing the sympathetic and parasympathetic nervous systems. So I would definitely know these because this would be a really easy test question to ask. Comparing and contrasting the sympathetic and parasympathetic nervous systems. So if we take a look at the word sympathetic, so that first part, sym, that means together, and path means to feel. So sympathetic got its name because you've got these widespread effects on your body and your whole body is feeling the same way, okay? So the sympathetic nervous system, when you stimulate it, you get these widespread effects, and they're longer lasting effects than the parasympathetic nervous system. Okay, so why do we get these widespread effects? Well, there's two reasons. The one has to do with this sympathetic chain pathway that we talked about before. So remember that we've got this preganglionic neuron starting in the spinal cord, and then it's going to synapse with the postganglionic neuron in this sympathetic chain right outside the spinal cord. And this preganglionic neuron has several collateral axons that will go up and down the sympathetic chain. So it doesn't just synapse with one postganglionic neuron, but it will synapse with many. It can actually synapse with, on average, like 17 postganglionic neurons. So you end up diverging and spreading out the signal and being able to talk to many different organs and getting them the signal that, hey, we need to fight or run away from, whatever the stressor is. So that'll give us some widespread effects. And then the other reason we get widespread effects is because the sympathetic nervous system also innervates the adrenal medulla and stimulates it to release the hormones epinephrine and a little bit of norepinephrine into the bloodstream. And those hormones go all over the body. And they will bind to receptors on sympathetic targets all over the place. And so that will also cause widespread effects throughout the body. And then the other thing with the sympathetic nervous system is we get much longer lasting effects than we do with the parasympathetic nervous system. And you know this because like maybe you were driving down the road and maybe another car almost hit you. And you get your heart pounding in your chest, and it takes a couple minutes for your heart to calm down and go back to the normal heart rate, doesn't it? Okay, so you've got these longer lasting effects, okay, from sympathetic stimulation of like your heart rate. It doesn't go back to normal right away. Okay, so the reason this happens, why we have these longer lasting effects, one is it takes longer to get rid of the norepinephrine at the synapse. And then two is you've got these hormones, epinephrine and norepinephrine, released into the blood by the adrenal medulla, and it's going to take longer to get those out of the blood and get rid of those. Okay, so while they're still in the blood, they're going to be having their effect and, causing the heart rate to increase until you get rid of those. Okay, the parasympathetic nervous system, how it got its name is not very original. Para means next to. So they just named it as the nervous system next to the sympathetic nervous system. And it's going to have, kind of the opposite effects of the sympathetic nervous system, rest and digest. And then also, it's going to have more localized effects and shorter-lived effects as well. So just the opposite of sympathetic stimulation. The reason we have more localized effects and we don't have these widespread effects is we have a lot less divergence. And that's again because of the anatomy. So remember that we talked about how the preganglionic parasympathetic neuron is really long, and then the postganglionic parasympathetic neuron is really short. So this preganglionic neuron is going to travel, to just about the target organ and then synapse, pretty close to that target organ. And so there's really not going to be a lot of collateral organs coming off here and being able to travel to other postganglionic neurons and communicating with other target organs. So there's really not a lot of divergence and spreading out of the message, and so it's just more localized effects. Okay. And then also the effects are going to be shorter lived. They don't last as long. And that's because the neurotransmitter acetylcholine that's used is much more rapidly destroyed at the synapse by the enzyme acetylcholinesterase, so it doesn't hang around as long.